Hello students and my dear friends, myself Madhu Vanja. In this session, we'll discuss growth, nutrition and metabolism of bacteria. These are the learning objectives. After completion of this session, you should be able to classify the bacteria based upon the nutritional requirements, describe the bacterial cell division, describe the various phases of bacterial growth curve. So here if you see the introduction, what are the minimum nutritional requirements for the growth of the bacteria? Like they are carbon, oxygen, nitrogen, hydrogen, and some of the inorganics also. So these are minimum nutritional requirements for the growth of the bacteria. To grow the bacteria, we have to provide all these nutrients in the form of artificial culture media. Next, nutritional types of bacteria. Based upon the type of nutrition, the bacteria, they are mainly classified into two types. The first one, autotrophs, and second one, heterotrophs. So what is meant by autotrophs? Autotrophs means they synthesize their essential metabolites from atmospheric carbon dioxide and nitrogen. Auto means a self. So they synthesize their essential metabolites from atmospheric carbon dioxide and nitrogen. Again, autotrophs, they are of two types. One is photoautotrophs and second one chemoautotrophs. So photoautotrophs, they obtain their energy photosynthetically. And chemoautotrophs, they obtain their energy by oxidation of chemical compounds. And next, heterotrophs. So what is meant by heterotrophs? So these are the bacteria, they are unable to synthesize their own metabolites. They cannot synthesize their own metabolites. They depends upon the preformed organic compounds. So if you see most of the pathogenic bacteria, what is meant by pathogenic bacteria? Disease causing bacteria. So disease causing bacteria are heterotrophs. So that's what they are, parasitic. So they depends upon the host body. Who is host here? Like we are only host here. So they depends upon the host body for the nutrition because they unable to synthesize their own metabolites. So they are evolved with the animal body. Why they are evolved in the animal body? Because ready-made nutrients are freely available. Next, microbial growth or bacterial growth. So what is growth in the microbiology? The growth in the microbiology refers to the increase in the number of cells. Always the bacteria, they increase in the number of the cells, but not in the size of the cells. Which means like one bacterial cell will be divided into two, two will be divided into four, 4, 8, 8, 16, 16, 32. Like that, the number of bacterial cells are increased, but they do not increase in the size. And next, bacterial cell division. So bacterial growth. So how the bacteria divides? The bacteria divides by the process called binary fission. So binary fission, it is a process of cell division. So bacteria divides by the binary fission. So how the binary fission takes place? Here, first of all, the bacteria, it feed upon the nutrients. So bacteria, it reaches to the critical mass in its cellular constituents and the cell division starts. First of all, the bacteria starts utilizing the nutrients. They reaches to the certain mass. They reaches to the critical mass, and after that only the cell division starts. So in this, first step is the division of DNA. Here the nucleus or chromosome, that is double-stranded DNA. The double-stranded DNA, it is divided into two single-stranded DNAs. The double-stranded DNA is divided into two single-stranded DNAs. So by this, the binary fission starts. And in the next step, the double-stranded DNA, again, they form the new complementary DNA strands. So in the first step, the double-stranded DNA is divided into two single-stranded DNAs. So these two single-stranded DNAs, they form the complementary DNA strands and they become the double-stranded DNAs. And these double-stranded DNAs, they are equally distributed between the two daughter cells. these are distributed into the two daughter cells, and a transverse septum is grown in the middle of the cell. Transverse septum is grown in the middle of the cell. So this is transverse septum start growing from the cell membrane, and all the cell wall, cytoplasm and cytoplasmic contents are equally distributed between the two daughter cells. And finally, these two daughter cells are separated. So this process is called as the binary fission. And in some of the bacteria, even after cell division, the bacterial cells are not separated completely. They remain partially attached. So that is the reason for the formation of chains, clusters. Okay. So here, if you see the clinical significance, so some of the bacteria, they are present as diplococci, like Neisseria species, pneumococcus, and others are present as chains, like streptococcus. And why this is streptococci, or why these clusters, or why this diplococci is formed? It is because of the non-separation of daughter cells after the cell division. The cells are not separated completely. So these morphological features, they are helpful in the identification of bacteria in the laboratory. Next, generation time. So what is meant by generation time? It is the time required for one bacterium to give rise to daughter cells under optimum conditions is called as the generation time. It is the time required for one bacterial cell to give rise to daughter cells under optimum conditions is called as the generation time. So most of the medically important bacteria, they have generation time of 20 minutes. Like if you take either E. coli or staphylococci, so they divide within the 20 minutes, which means for every 20 minutes, the number of bacteria will become double. And in case of Mycobacterium tuberculosis, the generation time is 20 hours. Means for every 20 hours, the number of bacteria will become double. So that's what it is slow growing bacteria. And in case of lepra bacilli, that is Mycobacterium leprae, the generation time is of 20 days. Means for one cell division, it will take 20 days of time. So it's a very, very slow growing bacteria. And what is the clinical significance here? So as we discussed here, most of the medically important bacteria, it has generation time of 20 minutes. So that's what the colonies are formed within 18 to 24 hours on the culture media. So when we inoculate the sample on culture media, within 18 to 24 hours, the colonies are formed. And in case of Mycobacterium tuberculosis, so generally the colonies are formed within the weeks. It may take around 6 to 8 weeks. And Mycobacterium leprae, it will take months to grow in the animals. So in the model animals, it will take almost months to grow the organism. In actually, the bacteria grows so rapidly. The bacteria multiply rapidly by the geometric progression, so that a single bacteria, it gives rise to the 10 to the power of 21 cells progeny in the 24 hours. But in actual, the multiplication of bacteria is arrested. Why the multiplication of bacteria is stopped? Because of exhaustion of nutrients. After certain period in the culture media, the nutrients are exhausted. And not only that one, here the accumulation of toxic products. The bacteria release the end products into the culture medium only. So they are the toxic products to the another bacteria. That's what the bacterial growth is arrested. Next, bacterial count. So here there are two methods are there to count the bacterial cells. One is total count and another is viable count. In the total count, we count the total number of bacteria in the specimen, whether they are the living cells or dead cells. So we generally count the total number of the bacteria. So this is done by using of counting chambers. So bacterial specimens, they are placed under the counting chambers, like Newbar chamber, and they are observed under the microscope. And in case of viable count, we measure only number of living cells. So how we measure the number of living cells? That is done by the dilution or plating methods. So in the dilution method, what we do, we take the several tubes which consist of liquid culture media, and they are inoculated with varying dilutions of sample, and viable count is calculated statistically from number of tubes, which shows the growth. And in case of plating method, so dilutions of the specimens are inoculated on solid media, either on the surface of plates or pore plates. So we count the number of colonies. So generally colonies are formed after incubation of around 18 to 24 hours. So then the colonies are formed. So each colony represents the clone of bacteria, which is derived from the single bacterial cell. Next, bacterial growth curve. And if you see the clinical significance of viable count. So viable count, it is helpful for testing of water. That is helpful for testing of water for presumptive coliform count, as well as even it is helpful in the diagnosis of urinary tract infections. And next, bacterial growth curve. So what is bacterial growth curve? When a bacterium is inoculated into a suitable culture medium and incubated, then the growth of the bacteria follow the definite course. So if we take a bacterial cell and if we inoculate into the new culture medium, then the growth of the bacteria takes place. So if we take the number of bacteria, the time on the x-axis and the number of bacterial cells on the y-axis, will get the bacterial growth curve. When bacterial count of such culture is determined at a different intervals and plotted in relation to the time, then the growth curve is obtained. So there are 4 phases are there in the growth curve. The first one is lag phase, second one log phase, third one stationary phase, fourth one death or decline phase. Like first phase is lag phase, second one log phase, third one stationary phase, fourth one death phase or decline phase. So in this picture you can see, so here we have taken the time on the x-axis and the number of bacterial cells on the y-axis, so that we'll get the bacterial growth curve. We'll discuss one by one. So the first one is lag phase. So what is the lag phase? So this is the immediate phase after inoculation of the bacteria. It is the immediate phase between the inoculation of bacteria and multiplication of bacteria. So after inoculation of bacteria into the new culture medium, the growth of the bacteria does not begin immediately. So in this phase, the bacteria it adjusts to the new environment. In the lag phase, the bacteria adapt or they adjust to the new environment, because we have taken the sample from some other culture media, and we are transferring them into the new culture media. So after transferring them into the new culture media, so in the new culture media, the bacteria just adjust to the new environment. So in this phase, necessary enzymes and intermediate metabolites are built up in the adequate quantities, which helps for multiplication to proceed. And in this phase, there is no increase in the number of cells, but there is appreciable increase in the number of cells, which means only a very less number of cells are only increased. So here in this phase, the bacteria adjusts to the new environment and it is ready for the cell division. And the duration of lag phase, it varies with the species and the nature of culture medium and the temperature. Next, the second phase is log or exponential growth phase. Second phase is log or exponential growth phase. So in this phase, we can see the rapid cell growth, that is exponential growth. So in the first phase, the bacteria adjust to the new environment, and in the log phase, after adjusting to the new environment, the bacteria starts multiplying, which means the population doubles for every generation, which means 1 will be divided 2, 2, 4, 4, 8, 8, 16. Like that, the number of bacterial cells are increased. So that's what we got the straight line. So here you can see the straight line, the straight line is obtained. And during this staining, during this phase, the bacterium is uniformly stained. Whenever we go for staining, so they are uniformly stained. And this is a better time to perform the gram staining. And in this phase, the bacteria, they are biochemically active, and it is the best time to perform the biochemical tests. Next, third phase is stationary phase. So after log phase, the bacterial growth is almost ceases. So almost it is stopped because here the death rate is equal to the rate of reproduction, which means the number of progeny cells which are formed, they are just enough to replace the number of cells that died, which means the number of cell birth is equal to the number of cell death. So in this phase, the cells start dying or the cells begin encounter environmental stress. So why the growth is arrested or why the cells start dying? It is because of lack of nutrients. So after certain period, so as the bacteria continuously utilize nutrients, so the nutrients are deprived and lack of water, and there is no enough space in the culture, in the culture media or in the conical flask, and metabolic waste, accumulation of metabolic waste, and oxygen is decreased, and the pH of culture medium is also changed. So all these are adverse conditions for the growth of the bacteria. And in this stage only, the endospores are formed. Next, last phase is death phase or decline phase or phase of decline. So in this case, in this phase, the bacterial population, it decreases due to the death of the cells. In this phase, the bacterial cells, they starts dying. So why the cells start dying? Due to the exertion of nutrients and accumulation of toxic products, and by the autolytic enzymes. So in this phase, there is decline in the viable count, but there is no decline in the total count. Why there is no decline in the total count? In the total count, we count both the number of living cells as well as the dead cells. But in this phase, the viable count is decreased. And next, morphological and physiological alteration of cells during the growth curve. What will happen at the end of the lag phase or in the log phase or in the stationary phase? So what happens in the end of the log phase? In the end of the lag phase, the bacteria have the maximum size. The bacteria, they utilize the nutrients, they reaches to the maximum size. This starts ready for the cell division. And in the log phase, the growth of the bacteria takes place, the multiplication of the bacteria takes place. In this phase, the cells are smaller and they stains uniformly. So when we go for the gram staining, the cells stains uniformly. And in the stationary phase, the cells are gram variable, which means they show the gram positive or gram negative. They show the irregular staining. And in this stage only sporulation occurs, and in this stage only they produce endotoxins. Why sporulation occurs? Because of the exertion of the nutrients and because of the adverse conditions. And in the phase of decline, so involution forms are common. So what are involution forms? Swollen and aberrant forms. Next, batch culture and continuous culture. So when a bacteria is grown in a vessel of liquid culture medium, that is a batch culture, after certain period, the cell division ceases. The cell division starts, the cell division stops, and the multiplication of the bacteria is stopped. Why the multiplication of bacteria is stopped? It is because of the depletion of nutrients and the accumulation of toxic products. Then how we can continue the culture? So we can continue the culture by replenishing the nutrients and removing of bacterial cells. So if we remove the bacterial cells, the dead cells or otherwise waste products, and if we replenish the nutrients, because of depletion of nutrients only the cells starts dying. So if we replenish the nutrients and if we remove the bacterial cells, so that we can continue the culture, that is called as continuous culture. So what is the purpose of this continuous culture? So this continuous culture, it is mainly used in the industrial as well as research purposes, where in case of vaccine production or in case of antibiotic production, so we use this continuous cultures. Next, bacterial growth in vivo. So what is bacterial growth in vivo? That is that is in the human body. So there is a significant difference of bacterial growth in the human body and artificial culture medium. So generally the bacteria multiplies in the host tissue, that is in the human body. So whenever they grow in the human body, the situation is intermediate between a batch culture and a continuous culture. So why there is intermediate in between batch culture as well as continuous culture? Because there is availability of nutrients is plenty. So it is host body is like rich in the like nutrients. So the source of nutrients plenty, but the defense mechanism of the body influences the growth. So that's what there is like equilibrium is maintained. Next, bacterial nutrition. If you see, the principal constituent of bacterial cell is water. So almost 80% of the total weight of the bacteria is the water. 80% of total weight of bacteria is the water. And as we discussed, what are the minimum nutritional requirements for the growth of the bacteria? They are water, source of carbon, nitrogen, and inorganic salts. So all these are minimum nutritional requirements. So these minimum nutritional requirements, they are supplied in the form of artificial culture media. And in addition to this, so some of the bacteria require like sulfur, phosphorus, sodium, potassium, magnesium, iron, and manganese in the small quantities. But major nutrients or macronutrients are like carbon, nitrogen, and inorganic salts. So these are all micronutrients such as sodium, potassium, magnesium, iron, manganese. And some of the bacteria are fastidious. Fastidious means they are exact in their growth requirement. So only in the presence of that growth factors only in the presence of that substances only they show the growth. So they require the special growth factors or bacterial vitamins. So if we do not provide that growth factors or bacterial vitamins, the growth of the bacteria doesn't takes place. So here, like what are the bacterial vitamins? So they are same like of the vitamins what we what we use, like especially they require the vitamin B group such as thiamine, nicotinic acid, riboflavin, folic acid, and vitamin B12. And these are essential for some of the bacteria. So which means in the presence of that in the presence of that growth factors only the growth of the bacteria takes place. So what is the purpose of what is the purpose of knowing this bacterial nutrition? So what is the clinical significance? So if you know the nutritional requirements of the bacteria, so you can provide these special nutrients and you can prepare the culture media so that you can easily isolate the particular bacteria. Next, environmental factors which affect the growth of the bacteria. So the first one is temperature. So here the optimum temperature for growth of the bacteria is 37 degrees centigrade. So what is optimum temperature? At which temperature the bacteria show the best growth, or at which temperature the bacteria show the maximum growth, that is called as the optimum temperature. So this optimum temperature is varies with the different species. So different bacteria, they have different optimum temperature, but most of the pathogenic bacteria, or most of the disease causing bacteria, they grow best at a temperature of 37 degrees centigrade, which is also like a human body temperature. Like that is a normal temperature of the human body. Here, based upon the temperature requirement, the bacteria, they are mainly classified into three types. The first one is psychrophiles or cryophiles. So they are cold loving microbes. Psychrophiles means cold loving microbes. Philic means loving. Psychro means cold. They are cold loving microbes, which means they grows in a temperature range of 0 to 20 degrees centigrade. The example, soil and water saprophytes. So water saprophytes, which feeds upon the dead and decay organic matter. Next, second one, mesophiles. So meso means moderate. They are moderate temperature loving microbes, which means they grows in a temperature range of 25 to 40 degrees centigrade. So majority of the pathogenic bacteria, most of the pathogenic bacteria, they are mesophiles. So they grow at a temperature range of 25 to 40. And as we discussed, they have optimum temperature at 37 degrees centigrade. They grow best at optimum temperature of 37 degrees centigrade, which is our normal body temperature. Next, thermophiles, which means they are heat-loving microbes. They grow in a temperature range of 55 to 80 degrees centigrade. Example, Geobacillus stereothermophilus. So previously, they are called as Bacillus stereothermophilus. So what is the clinical significance? Why you should know the temperature requirement? So most of the pathogenic bacteria, they die quickly in between 55 to 65 degrees centigrade, and the spores are killed in between 100 to 120 degrees centigrade. So this will help you for the sterilization. So you'll know the sterilization, which means destruction of microorganisms, how we can use heat, either autoclave or hot air oven. And some of the bacteria, they die quickly on exposure to the low temperatures, Haemophilus influenza. Haemophilus influenza, that is it cannot survive in the cold environment. And even the bacterial cultures, they are stored in the refrigerator. So that is in between 3 to 5 degrees centigrade, or in the deep freezer, that is in minus 30 degrees centigrade, or to minus 30 to minus 70 degrees centigrade. That is for maintaining the stock cultures. Next, pH. So most of the medically important bacteria, they grows in a pH range of 7.2 to 7.6. And very few bacteria, they grow at a pH of 4.0, that is acidic pH. So if you see lactobacilli and vibrio, so lactobacilli, it can grow in the acidic environment, that it is pH of 4.0, and vibrio cholera can grow in the alkaline environment. So that's what in the preparation of culture media, especially for vibrio, we maintain the alkaline pH. Next, osmotic effect. So bacteria, they able to withstand wide range of osmotic variations, external osmotic variation. Why they are able to withstand wide range of osmotic variation? Because of their cell wall, because they are a tough cell walls. But sudden exposure of bacteria to the hypertonic solution, it leads to the plasmolysis. So what is plasmolysis? It leads to the shrinkage of the protoplasm, and sudden transfer from concentrated solution to the distilled water leads to the plasmoptosis. So what is plasmoptosis? That is a rupture of the cell wall. Next, oxygen. So based upon the oxygen requirement, the bacteria, they are mainly classified into two types, aerobes and anaerobes. Aerobes means they require oxygen for their growth. Anaerobes means they grow in the absence of oxygen. And if you see the word obligate aerobes means strict aerobes, which means strictly they grow in the presence of oxygen. Example, if you see Pseudomonas aerogenes, it is a obligate aerobe. Next, facultative anaerobes. What is meant by facultative anaerobes? Ordinarily, they are aerobes. Generally, they are aerobes, but without the oxygen also, they can be grown. And if you see, most of the pathogenic bacteria like Staphylococcus, Salmonella typhi, they are facultative anaerobes. Next, microaerophilic bacteria. What is meant by microaerophilic bacteria? They require minute amount of oxygen, or they grow in the presence of traces of oxygen. Like if you see Campylobacter and Helicobacter pylori, they are microaerophilic bacteria. Next, obligate anaerobes. So what is meant by obligate anaerobes? Strictly they grow under absence of oxygen. Strictly they grow in the absence of oxygen. Even if minute amount of oxygen present, or even if traces of oxygen present also, they cannot be grown. Example, Clostridium tetani. Next, moisture and desiccation. Here, moisture is essential for the growth of the bacteria. So without the moisture, the bacteria cannot be grown. So drying has varying effects. If you see Treponema pallidum and Neisseria gonorrhoeae, they die quickly on exposure to the room temperatures. On exposure to the higher temperatures, they die quickly because they are very delicate bacteria. So that's what during the transportation to the laboratory, they are placed in the transport media. So transport media, they do not allow the bacteria to multiply, but they maintains the viability. So why? Because this Neisseria gonorrhoeae, if they are exposed to the room temperature, they will die. So that's what we'll place them in the transport media. And if you see Mycobacterium tuberculosis and Staphylococcus aureus, they survive for several weeks. So next, drying in cold and vacuum conditions is called as lyophilization. So what is the purpose of lyophilization? It is used for preservation of microorganisms. For long-term storage of microorganisms, we use the lyophilization. Next, carbon dioxide. Even small amount of carbon dioxide is required by all the bacteria. So we need not to provide specifically carbon dioxide because it is available in the atmosphere, or this carbon dioxide is produced endogenously as a product of cellular metabolism. So the bacteria itself, it produces the carbon dioxide as an end product of the cellular metabolism. But if you see Brucella abortus, it require higher level of carbon dioxide, that is 5 to 10% of carbon dioxide, which is called as capnophilic bacteria. Next, light. So most of the bacteria, they grow well in the darkness, except phototrophic species. Why phototrophic species? They do not grow in the darkness because they require the sunlight. And most of the bacteria, they are sensitive to the ultraviolet rays and other radiations. And photochromogenic mycobacteria, they produce pigments on exposure to the light. They produce colors on exposure to the light. Next, mechanical and sonic stresses. So even though the bacteria have the tough cell walls, so if we add glass beads to the culture, if we vigorously shake it, the cell walls are ruptured, and even if they are exposed to the ultrasonic vibrations also, they can be ruptured. Next, bacterial metabolism. So what is meant by metabolism? So metabolism, it is a series of changes of substance like a carbohydrate, protein or fat within the bacterial cell from absorption to elimination is called as the metabolism. So what happens for this carbohydrate, protein and fat in the bacterial cell from the absorption to the elimination is called as the metabolism. And if you see aerobic bacteria, they obtain energy through the oxidation, which involving the oxygen as the ultimate hydrogen acceptors. And in case of anaerobes, so anaerobes, they do not require oxygen. So other than the oxygen, they use hydrogen acceptors. Anaerobes, they use hydrogen acceptors other than the oxygen. And oxidation, what is meant by oxidation? So in case of aerobes, hydrogen acceptor is oxygen. So the carbon and energy source may be completely oxidized to the carbon dioxide and water. And the conversion of adenosine diphosphate, that is ADP, to the adenosine triphosphate ATP, it provides energy in this process that is called as the oxidative phosphorylation. And a small amount of hydrogen peroxide is produced in this process, is disposed of by the enzymes such as catalase or peroxidase, which are present in the aerobes. And next, fermentation. So in case of anaerobic bacteria or in case of anaerobic respiration, hydrogen acceptor compounds are nitrates or sulfates instead of oxygen. So instead of oxygen, hydrogen acceptors are like nitrates or sulfate. In fermentation, a series of oxidoreductions occurs in which carbon and energy source act as both electron donor and electron acceptor. And what are the end products of fermentation? What are the end products of fermentations? Acids, gases, and alcohols. Next, oxidation reduction potential or redox potential. So what is oxidizing agent? It is a substance which accepts electron while reducing agent is able to part with the electrons. The oxidizing or reducing condition of a system is indicated by the net readiness of all components in that system to accept or to part with the electrons. This is the end of the session. So what is the summary? Here, what are the minimum nutritional requirements for growth of the bacteria? Like carbon, hydrogen, oxygen, nitrogen, and some of the inorganic salts. And bacteria divides by the process called as binary fission. And bacterial growth curve has 4 phases: lag phase, log phase, stationary phase, and death phase. And what are the factors which affect the bacterial growth? That is temperature, carbon dioxide, oxygen, and moisture desiccation. Thank you.